Okay, so good morning, everyone, and welcome back. This is the course on bioethics at the beginning of human life within the program of the Master in Bioethics. And today's lecture is going to be on stem cell research and cloning. So I promise it will be an exciting one. We begin, as always, with a little prayer in the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we thank you once again for your many blessings, for the gift of life, for having us here today, once again, to delve deeply into nature and our human nature, especially as we consider our individual origin, ontogenetically, as we say, Lord, when we begin, each one of us, at that time of fertilization, and then all the bioethical issues that surround the beginning of human life today in our complex society. We ask that you send us your Holy Spirit to illumine our hearts and minds, always to discern your will in our lives and to live according to your will. We thank you, Lord, for your many blessings, and we pray in a very special way for those who are in most need of your divine mercy. In the name of Christ, our Lord. Amen. Okay, so any questions or comments from previous lectures?
As I said to the
Yes.
the last lecture, I had so much information.
There is, yes.
I left off a whole lot on some of it.
Sure.
But I have 2 more pages that I kept back. And I did do, so I put them,
Sure.
when I get back.
No problem.
So much.
Yeah.
So much info.
And just for the record, that's Deacon Lewis, and he gives me 2 pages, but it's a single space, all one sentence after another, no break in the paragraph. So it's 2 solid pages, 1 full sheet of wording and sentences, okay, that are also compressed. You have good summaries, Deacon. You know how to summarize well.
The point why I do that way, because if I'm going to teach this, I'm going to be teaching directly on that paper.
Yes.
So it's already there. Yeah.
You know exactly. You have it. So it's all in the preparation, right? Yes. As teachers, we know that the quality of the lecture is only as good as the preparation. And it's also providential that we have a number of teachers in this particular cohort. So we know that I always say that the best teacher is a student, right? We're always learning. And I say the best student is also a teacher. So we have this beautiful and great capacity to learn and then to communicate what we learn, which is fantastic. In fact, essential really when you think about it, because none of us are born with the full knowledge, right? We are born ignorant. And each generation has to be taught again and again, as we know from teaching elementary, high school, each generation has to be taught again. But a great privilege, okay? Also, with regards to the lectures in general and the summaries, I'm always looking for the big picture, right? So I tend to go into a lot of detail just to try to cover the basis as much as possible. And being that our cohorts are varied, people have very different backgrounds, all the way from theological background to scientific background. So I'm always trying to be as thorough as possible in each lecture, but always look at the big picture, okay? And that's the idea. The background is so that when you do get into these discussions, my father, when you get into these teachings and discussions and dialogues, hopefully, you can speak with competence. That's the whole idea, okay? So it's not always that we have to use every single argument or every single detail in the topic, but it's important to have it as background so that you can pull it up. Okay, so we have been looking at mostly the biology of the beginning of human life, so as to ground it really at what we call completed fertilization syngamy, when we have at least those 3 levels of unity, of linking, at the cellular level between the two gametes, at the nuclear level between the two pronuclei, right? And at the chromosomal level with the two sets of chromosomes, the 23 chromosomes in the case of the human, the 23 from the father, the 23 from the mother, to give the full complement diploidy, what we call restoring diploidy, right? And that's what fertilization is all about at the biological level for the next generation. So imagine just for a moment, just let's back up for a moment and look, as it were, as an eagle's view of what's going on, okay? In those two gametes, in those two cells, the egg and the sperm, that fuse in the process that we call cellular fertilization, a new generation starts. The entire package of all the characteristics that make up the human body, or any body of any species that reproduces sexually, is passed on to the next generation, okay, in just those two microscopic cells. It's amazing, we say in scientific language, the elegance of that mechanism, the elegance of that mechanism of sexual reproduction. And it's not only that, because asexual reproduction can do that even with a single cell, right? Through mitosis or just the other types of acellular reproduction. So, but the great advantage is what? Above and beyond asexual reproduction. For sexual, what's the big advantage? Being that it costs twice as much energetically, there's an expense of energy. What is the big advantage for the next generation?
It's getting all these strong genes.
Yeah, okay, so strong genes, meaning there's going to be what in the next generation? Diversity, variation, but variation within the species. So you really get the both worlds. The next generation, okay, the children of human are still human, meaning that when they become reproductive adults, they can produce their own offspring who are also human, right? So the integrity, if you will, of the species is maintained as a whole, in the number and the quality of the DNA, of the genes there, but at the same time, there's variation introduced. So there's variation within the species, okay, which allows for selection to happen, selective pressure. And again, selective pressure continuously is purifying the species. And so these mechanisms are set in place in nature that have evolved over millions of years. But if we go back even further, abstract even more, all right, to the bigger picture, these laws of nature, right, like selection and so forth, are mechanisms that have to occur when the environment is proper. That's why we say nature never forgives. A weaker organism, statistically, is going to be selected out, okay, especially when the climate changes somewhat. All right, when the conditions become a little more harsh. If the conditions stay stable, you know, even the weaker tend to survive. But when the conditions become a little harsher, the weaker tend not to survive. And that is really a good thing for the species as a whole, because that species continues to get stronger and stronger, and to the point where enough variation can happen into speciation, dipping a little bit into last semester's course. But the elegance of all this, and those laws had to be established at some point in its very origin, which is the whole functioning of nature and the entire universe, the laws by which matter and energy are locked into, if you will. So that's pointing to the creator of those laws. Okay, nature did not invent itself, as it were, because one of the laws of thermodynamics, right? That nothing is created and nothing is destroyed, everything is transformed. Okay, so coming back in, landing that eagle back onto Earth to our course today, after we establish the logical conclusion and the biological fact that the life of a new individual of whatever species begins at fertilization. Now we move forward. We looked at early embryonic development and the stages of development there. It's a continuous process of differentiation, of cellular specialization, cellular differentiation. into all of the organ and systems that we have in our body. And it's considered that we have approximately 220 cell lines, what are called cell lines. In other words, remember at gastrulation, right? We have the 3 germ layers. What are those 3 germ layers that form? Ectoderm, mesoderm, and endoderm, right? Derm is a reference to layer or skin in the loose sense of the word, but layer. And ecto, meso, and endo simply where they are located physically in that little embryo that is beginning to differentiate. From those 3 germ layers, all of the organ and tissues develop, okay? And it is established by scientists that all of our organ and tissues really are made up of about 220 cell lines, okay? Cell lines which go into specific organs and tissues. So for example, neuronal cell lines, there'll be several, 2 or 3 neuronal cell lines, I don't know exactly how many, but we can look at cells in the blood, for example, white blood cells, RBC, red blood cells, RBCs, that could be a cell line. White blood cells that have a different function. White blood cells have the function of the immune system, right? That could be a different cell line. The platelets have a 3rd function in the blood. Platelets form the scab for healing, okay? So those could be different cell lines. And our bodies are made up of about 220 cell lines. The idea with stem cell research, because we're going to get into this today, is to develop these cell lines in the lab and to have what is known as immortalized cell lines, to have the 200 plus cell lines, human cell lines immortalized in the lab for replacement of tissue that is injured or becomes ill and so forth. That's kind of the ideal, one of the goals for stem cell research. It's a very ambitious goal, and there's a lot of money put into this, and there's also a lot of politics involved in it because it's sophisticated technology, and with sophisticated technology comes economics, and with economics come politics, right? And so these 3 legs of the tripod, technology, economy, and politics, are intertwined in a complex dance. So bioethics is seeking to keep the politicians honest with our money, especially when we talk about public body, the funding, which is the big bucks. Okay, what else? So we looked at a couple of issues last class that are already, last lecture, that have a big social impact. One is abortion, the other one in vitro fertilization. You can see that they're heavily influenced by technology, and yet they, one way or another, they attack, they offend the human embryo in his or her earliest beginnings. And so these are really, at the end of the day, highly offensive procedures, bioethically speaking. Okay, hopefully I gave you the biological and the theological grounding for that, including the gift of a human soul when precisely we begin our biography, our individual history. Okay, so into stem cell research, actually 2 aspects. We'll look at stem cell research first, and then cloning, and specifically human cloning. I don't know if you saw in the news just last week, I tell you, not a week goes by without a bioethical issue, right? Human cloning, or not human cloning, but the precursor to human cloning. Anyone see the news last week about some cloning? No? China, the monkey, they cloned the monkey, all right? And the idea is that that's the precursor to human cloning, which actually has been done experimentally already in a few labs that we know of, but not carried to term. We'll get more into the details about that, but turns out that now for cloning, China was big news last week, was able to clone a monkey, which is an experimental monkey in the lab, but this monkey was in fact manufactured through cloning, okay? And monkey being a mammalian, being a primate, is essentially the same technique as human cloning, biologically speaking, technologically speaking, okay? But not ethically speaking, right? Okay, so let's look at stem cell research first. And what do we have? We have to make distinctions. I think I quoted Aquinas for you at some point, even though he believed in the late harmonization, because sadly he had a very defective biology back then. Essentially, the biology was very simplistic and not scientific enough to understand fertilization or syngamy or anything like this. But aside from that issue, that intuition of delayed harmonization, Aquinas was a very good theologian, and we call him the great systematizer. In other words, he's the one that systematized, put together a system for doing theology in our Catholic tradition back in the Middle Ages, 12th, 13th century, and he wrote manuals. The famous manual that he wrote was known as the Summa Theologica, which is volumes of how to do theology. Today, it takes time to reflect upon the writings of Aquinas in the Summa, and yet the Summa was intended to be the seminarian manual, basically the manual for seminarians at his time in the Dominican order, being that he was a Dominican. Okay, so all this to say that Aquinas had a little phrase, at least attributed to him. He would say, never deny, seldom affirm, and always distinguish. Okay, so a lot of theology and moral theology and bioethics, which in a sense is applied moral theology, is about making distinctions, right? For example, typically the question when it's presented, especially by the secular environment and definitely in secular media, the question is too simplistic, okay? For example, people will ask, or a reporter may ask you, well, is the church for or against stem cell research? Is the church for or against stem cell research? Well, that question is loaded with assumptions. First of all, the church, we assume they're talking about the Catholic Church because there are many other churches existing today in the world, but they're typically talking about the Catholic Church, right? Okay, stem cell research. Well, it depends. Frankly, that question, just simplistically how it's stated, is the church for or against stem cell research? I can say, honestly, the church is not only not against stem cell research, the church is actually in favor and is actually doing stem cell research. Oh, the church is doing stem cell research, yes. Hospitals that are teaching hospitals for medical schools, so there are medical schools that are Catholic medical schools, right? Georgetown, Loyola, Chicago, Fordham, Tulane, many Catholic universities have a medical school, and typically these medical schools are associated with the Catholic hospital, which is a teaching hospital, just like the Young Hospital is the teaching hospital for the University of Miami Medical School. And in those Catholic hospitals, there is stem cell research going on. You know what? That stem cell research is adult stem cell research. Okay, and so we have to make clarifications and we have to distinguish and clarify. So there are two levels of distinction when we talk about stem cell research. The first level is whether we're talking about human stem cells or non-human stem cells. That makes a big difference, first of all. Just like when we talk about the zygote, we talk about a human zygote, we talk about a non-human zygote. We can and we may experiment with non-human zygotes as long as there's a worthy cause, typically for medicine and advancement of the research of basic knowledge, but we may not experiment with human zygotes because we cannot obtain consent, for example, from a human zygote, being a human person. Okay. So the first level of distinction when we talk about stem cell research is, are we talking about human stem cells or non-human stem cells? So it's a big distinction, big difference, ethically speaking, bioethically speaking, and also biologically speaking. That's necessary, but not sufficient. We have to go deeper. Okay, well, we're talking about human stem cells. Well, we still need to make a further distinction, which is a big bioethical distinction, on the analysis, whether we're talking about adult stem cells or embryonic stem cells. Okay, so we've established that we're talking about human stem cells. Okay, the reporter is asking about human stem cell research. Yes. Now, are you asking me about embryonic stem cells or adult stem cells? See, the levels of distinction, just to be clear. Oh no, I'm talking about human embryonic stem cell research. Okay, now you've made the question specific enough to give you an answer. And the bottom line answer is no, may not do research or experimentation on human embryonic stem cell research. Why? Because it kills human embryos, bottom line. If we had a way of obtaining human embryonic stem cells that did not kill the embryo or significantly endanger their life, we could proceed with that kind of research. But the way that the human embryonic stem cells are obtained today, it involves the killing of the human embryo. They talk about destroying the human embryo and so forth, but it's really killing because the human embryo is alive. If the human embryo were not alive, we wouldn't be interested in the stem cells, right? Okay, so let's see where these human embryonic stem cells come from. They come essentially from the ICM. So we go back to the early embryonic development, the blastocyst stage, third stage, right? Zygote morula blastocyst is the first level of cell differentiation. We have at the level of the blastocyst, an inner cavity forms in a little ball of cells. And then there's going to be the outer layer of cells, which is called the trophoblast. And the trophoblast will become the placenta with the little embryo implants. And then there's that inner mass of cells inside that ball, inside that cavity. There's an inner cell mass, the ICM, which will become the embryo proper. And we even saw videos and a spectacular ultrasound imaging, MRI imaging of live human embryos developing from the ICM. So to obtain, and these are the embryonic stem cells, okay? These are the embryonic stem cells. To obtain them, this embryo, this blastocyst, has to be destroyed, has to be dismembered, the cells have to be disaggregated, and the ICM pulled out and further desegregated into a Petri dish that will have nutrient medium that is compatible with life, the nutrients that the ICM needs for survival. And this, of course, these Petri dishes, or the kind of glassware, is kept in an incubator at 37 degrees centigrade, which is body temperature, and so forth. So you can see that the bottom line is that the human embryonic stem cells come from the ICM, and the ICM is destroyed. The embryo, the integrity of the embryo is destroyed as such, killing, in effect, killing the human embryo. So that's the big opposition of the Catholic Church, and of that should be of any scientist and any person in society that has a conscience, to know that this human embryo is indeed human with a soul and with potential for further development. If we're left with embryo alone in his or her proper environment, meaning in the womb of a mother, within 9 months, this embryo should turn into a normal baby, like you and I. Okay? So that's the big issue here, is that to obtain human embryonic stem cells, the human embryo has to be killed. Okay, now, why are scientists intent, some scientists intent on going after the ICM, on these human embryonic stem cells? Because they are pluripotent. So let's talk about cell potency. Differentiation, cellular differentiation, means that as the cells grow and develop, they also specialize. They go from a general state into specialized tissues of our bodies, okay? And that process is called cellular differentiation. As cells grow and develop, they differentiate. They become specialized into the different organs and tissues of our bodies, okay? Precisely into the 220 cell lines that I was talking about earlier. However, if we go backwards in time, all these differentiated cells, the trillions of cells that we have in our bodies, making up the dozen organ systems that we have, you know, digestive system, reproductive system, nervous system, etc. If we go back in time, you know, a few decades, we go far enough, we go to the very origin, to the time that each one of us was a zygote, we start as one cell. So that cell is undifferentiated. That cell is the most plastic possible. That cell will eventually, through mitosis, become every cell in our body. So all of our cells, you can see, are daughters and granddaughters and great-granddaughters of the original mother cell, which was the zygote, right? So that cell is called, that one zygote is called totipotent because it has the full potential. Toti reference to full potential to become every cell in the body. In fact, at the level of the zygote and the morula, okay, at the level of the zygote and the morula, they can also become the placenta and the umbilical cord. And so they are truly totipotent, okay? So bottom line, the zygote and the morula are known as totipotent because all of the organs and systems, all of the cells in our body are going to come from these original cells. That's why also I mentioned that the blastomeres, what is a blastomere? A blastomere is a single cell of the morula, right? And it's also a single cell of the blastocyst, by the way. So it's unfortunate there's no real distinction here at that level of the name blastomere. But the morula blastomeres are also totipotent, to the point that if I take out a single blastomere from a morula, okay, that single blastomere can revert back to a zygote and start a new individual, an entire new individual. In fact, that's something of what happens in monozygotic twinning, okay? Because remember, the zygote can become a twin, a monozygotic twin at the two-cell stage, right? The two-cell stage, which is the first mitotic division, of the zygote, you have two blastomeres. Each one of those two blastomeres, if that separation is a radical separation, identical twins, right? Monozygotic twins. Okay, so it's all coming back. All right, so the morula and the zygote are totipotent. Totipotent cells, they have the full potential to generate all of the organ systems of the body. Once we get to the blastocyst and the gastrula, okay, sequentially, what happens at the blastocyst? What's the most significant biological event from the morula to the blastocyst? There is cellular differentiation, the first level of differentiation. Remember, the blastocyst has two different types of cells. Okay, they are different from each other, so there's cellular differentiation. One type of cell makes up the ball itself, around the ball, okay, the outside of the ball, and that's called the trophoblastocyst, the trophoblast. The other little mass of cell inside is the ICM, just unimaginatively called the inner cell mass, and the whole embryo comes from here. So there are two different types of cells, you see? There's the first level of differentiation. So that the blastocyst will become the placenta and not the embryo proper, and the ICM will become the embryo and not the placenta. Okay? So this first level of differentiation means that neither the trophoblast nor the ICM are any longer totipotent. They are now pluripotent. Okay, so they've lost some potency. They're somewhat restrained now. They're getting channeled further and further into a niche, if you will, a particular cell line or a cluster of cell lines. The next level of differentiation, and also, by the way, the gastrula, in other words, the three germ layers, ectoderm, mesoderm, and endoderm, are also considered pluripotent, okay? Pluripotent. But then when further differentiation happens, as the embryo begins to develop his or her organ systems, okay, then those cells are becoming more and more specialized into particular cell lines. They lose their pluripotency, and now they become multipotent. So they're further specialized. See? By the time we get to the little embryo about 1 inch long, remember I said all of the organs are formed, all of the main vital organs are formed and in place, but in miniature because the embryo as a whole is about 1 inch long. Okay, but all the organ systems are there. Okay, those are multipotent or even fully differentiated cells that form those organ tissues in the little embryo. And at that point, just to finish that sentence, it's mostly cell growth. The cells are mostly fully differentiated by that point, within 8 weeks of embryonic development, of pregnancy. And then at that point, it's mostly growth in size. So mitosis, again, the liver, which is tiny, has to become huge in comparison. The heart, which is also tiny, has to become large, and so forth, to maintain all the function of the bigger body, all right? And so it's mostly cell growth until birth, when the cells are more and more differentiated. And so significantly, the embryo changes name to fetus, which in Latin means tiny bean, foetus, tiny bean. John.
At this stage with the yolk sac, what is that called when it's at that point?
Okay, so yolk sac, we're talking about the blastula, the gastrula. This is the gastrula. Is still pluripotent. So that's why I put them here. Okay. Pluripotent. All right, so you got both at the blastocyst and at the gastrula stage, the cells are still pluripotent. Because, for example, all of the organ systems will come from these 3 germ layers. But we make some distinctions, right? The ones that are well known, for example, for the ectoderm is going to be the integumentary system known as the skin, which has ectoderm, has dermal layer, and then also the nervous system through that process of invagination. Whereas in the mesoderm, it will be the middle organs, mostly the digestive system and other organs in the center of the body. And the endoderm, typically muscle and the skeletal system. Okay. So you see they're becoming more and more differentiated, but not fully differentiated yet. So globally, we can talk about 4 stages of potency or cellular differentiation. And what are the 4 stages? Beginning with totipotent, to pluripotent, to multipotent, to fully differentiated. Okay? This is significant. This is important because we're talking about the quality of the cells. And the embryonic stem cells have what kind of potency? Embryonic stem cells, where do they come from? They come from the ICM, right? Therefore, they're pluripotent. Okay, now I'm telling you in anticipation that for actual clinical healing in the lab, in the clinic for human beings, multipotent cells are better than pluripotent. Sounds contradictory, but you'll see the reasoning, okay? Just in anticipation. That's why adult stem cells are better than embryonic stem cells, clinically speaking, aside from the ethical issues. That's providential. Okay, so 4 levels of cell potency: totipotent, pluripotent, multipotent, fully differentiated as the cells develop. And this is a one way. In nature, there's no reverting. In the lab, this process has been able to be reverted in the lab, but not in nature, okay? Let's look at adult stem cells. Where do adult stem cells come from? Well, the word adult is significant, right? To distinguish it from embryonic. But adult, we actually have to put it in quotations, if you will, because it's anyone who is born. So adult is to differentiate these stem cells from the embryonic stem cells. In other words, before birth or after birth. We know that the embryonic stem cells, typically they come from the ICM, so early on, the blastocysts. But adult stem cells, anyone who is born, actually a baby, a born baby, has adult stem cells. And so does the child, so does the teenager, and so do all of us, and even old people. We all have stem cells in our bodies once we're born. For example, if I cut myself, that cut will heal, hopefully, the normal process, from the inside out. The level of the dermis, even down to the adipose tissue, which is also known as fat, there are stem cells in there that generate new cells to heal that wound, okay? And those are stem cells. I'm born already, so those are, by definition, adult stem cells. But like I say, from babies forward, we all have stem cells in our bodies, in different parts of our body that are there waiting. They're called quiescent. They're quiet. They're kind of in an rested state until they're needed, if they're needed, because of injury or illness. And it's part of the repair mechanism, not at the DNA level, but at the cellular level. It's repair mechanism at the cellular level, okay? So there are stem cells in our skin, like I mentioned, in our fat tissue. I tell the women, next time you have a liposuction, save your stems, save the fat tissue, because there are stem cells in there. There are stem cells in the pulp of baby teeth. You know how we shed our teeth normally once in our lifetime, and the pulp of baby teeth have stem cells, adult stem cells, that are neurologically predisposed. Okay, so they're multipotent in the neurological line. Hair follicles have stem cells, adult stem cells. We lose the hair and a new hair will come out through the hair follicle. The classical one that was not identified as a stem cell transplant originally, but has been going on for decades really, is bone marrow. Bone marrow, which is where red blood cells and white blood cells are produced inside the bone, right? Especially the long bones in our body, the femur, and also the ribs are known for producing a lot of blood cells. And the marrow, which is a dark, deep reddish color, that bone marrow has adult stem cells. And bone marrow transplant has been going on for years, right? Okay. So all those are examples of adult stem cells. Even stem cells in the brain have been identified in a region called the hippocampus, which is toward the center, the very center of the cell, okay? The hippocampus, which apparently can be stimulated by oxygen to regenerate. And that's a big to-do, because as the cells are more specialized, the less potency they have, the less capacity for regeneration that they have. And the most specialized cells in our body are the neurological stem cells, the neurological cells. And that's why nervous injuries to the spinal cord and so forth are so difficult to heal, because generally nerve cells do not regenerate. Now they're talking about that they regenerate very, very slowly. So we have different sources of stem cells in our bodies. The liver is another one. About 1/3 of the liver could be excised, and that liver will regrow. So the liver also has stem cells in them, and they're predisposed, and typically they are multipotent. So you see, they're not pluripotent like the embryonic. They're more toned down, if you will. They're more specialized into particular organs or cell lines. But I'm telling you that that is actually a benefit. And you'll see why in a minute, the biological reasoning why. Now, one difficulty with the embryonic stem cells is that necessarily they come from another individual who was the human embryo that was killed to obtain those embryonic stem cells, right? So the embryonic stem cells necessarily come from another individual. When they are transplanted into my body, myself being ill with something or injured and I need stem cells, okay, if they are embryonic stem cells, They're human, but they come from another individual. So there's going to be immediately an immune rejection to those embryonic stem cells, which means that the patient now has to be put on what we call immunosuppressors, just like a transplant, all right? The patient has to be put on immune suppressors so that the patient will not reject the embryonic stem cells. I'm talking about a hypothetical case. That's a negative because now the patient not only is sick or injured, but now the patient is also on immunosuppressors, more susceptible to infections. The good thing about adult stem cells is that they can be autologous. In other words, they come from my own body. They're taken out from one part of the body and injected into another part of the body. But because they are my own stem cells, then there's no immune rejection. They're compatible. They are immunocompatible, okay? And that is called autologous, from the same body, from the same person. And there are many, many successes that have occurred also in the adult stem cells, in contrast with the embryonic stem cells. Now, typically, embryonic stem cells, we have yet to have a single successful trial, clinical trial in a human with embryonic stem cells. They have been studied extensively in lab animals, typically the mouse, and the classical experiment for embryonic stem cells is a mouse that is injured. And then let's follow the scenario. You take a female mouse or female rats. Rats are easier to work with because for certain reasons they're larger. Okay, so it depends. If you want more litter and so forth and faster growth rate, you use mice and the lab mouse. But if you want to look at organs, regeneration and so forth, you use the rat because they're larger, just physically larger. Okay. What happens? You take a female rat who is pregnant, or actually not pregnant, you have to do let me follow the scenario mentally. You need to do in vitro to obtain the blastocysts of the rat. So you take a female rat, hypo-ovulate her, take out the eggs and fertilize, do the in vitro fertilization, do IVF on the rat, so you also have blastocysts from that rat. Okay, so at least the blastocyst is going to be 50% compatible. You're trying to get as compatible as possible to the rat's immune system. So you have a rat, and you have the blastocyst. You can use the male of the rat also, who fertilized the eggs. So you find 50% compatibility on both sides. Now you injure that rat, okay? And the typical injury, let's say, is to sever the spinal cord. So the rat becomes either paraplegic or quadriplegic, depending on when you do the injury on the spinal cord. But they become disabled either from the neck down or from the thorax down. Okay, these are experimental things that are used. They can be justified ethically. Let me address the issue. If there is a promise for curing injuries to humans, spinal cord injuries, and the typical injury is in the military, but not necessarily. It could happen. I have a friend of mine who was doing exercise one day, fell off the rack, and he severed his spinal cord, and he's been paralyzed. He's quadriplegic from the neck down for decades now. So it can happen, you know, an accident can happen. At any rate, now you have this paralyzed rat, okay, and you have blastocysts from that rat. So you take the ICM and you develop in the lab stem cells, embryonic stem cells from the blastocyst of the rat. Could be, like I say, either the male or the female, because each one is going to be 50% compatible with those embryonic stem cells. Now you do the transplant, you literally take those stem cells, embryonic stem cells, and inject them directly into the site of injury to see if those stem cells will regenerate the nerve cells that are needed, okay? So the first issue you have to deal with is the immuno incompatibility, because the other 50% that those stem cells, those embryonic stem cells, are only 50% compatible, right? So you have to put those rats on immunosuppressants. You can do that. You tone down their immune system. You keep it in a controlled environment, sterilized. Sometimes they've also developed nude rats. Nude rats don't have any skin, they have skin, they don't have any hair, because hair carries a lot of bacteria and so forth, fungi, so it could infect them. Anyway, you have all the control conditions, right? What will happen? What is a typical classical scenario when we do that kind of experiment with embryonic stem cells that are pluripotent, right? So theoretically, they are the best to heal because the nervous system is going to come from those pluripotent cells, okay? The nervous system is going to come from there. You know what happens typically in the site of injury where the embryonic stem cells were injected? Typically, they grow tumors. They grow tumors, cancerous tumors. Why? Because those pluripotent cells actually have too much potential. They have too much potential. You see? It's like the whole little embryo wants to grow there. They have too much potential. So, whereas theoretically, what we call the gold standard is the embryonic stem cells for healing any organ of the body, because all of the organs of the body come from the ICM, okay? Theoretically, they are the gold standard. But in practice, clinically, in the lab, what happens is that those pluripotent cells just have too much potential, and the whole darn little embryo wants to grow there, and typically they grow tumors. Now you translate that into a human clinical trial. First, you have the issue of the immune system, which again can be done. You know, transplant patients, they live with immunosuppressors, and they just have to be careful about infections. But now the injured person is going to develop tumors as opposed to heal. So it's a big negative in the practice. So it's not about the theory. Theoretically, yes, they're pluripotent, we understand, we get it. But in practice, that pluripotency is too much. Better, multipotent. In other words, they're already lined up in a particular cell line or cell pathway that is better because their potency has been toned down. See? And that's why adult stem cells are actually better in the practice. And so I make the analogy, simple analogy of you have a transistor radio when there used to be transistor radios around, okay? And they need, they just need a AAA battery, a small battery. But now I have this AAA battery that has 200 volts in it. It doesn't have 1.5 volts, it has 200 volts. And I put that battery into the radio and the radio fries, okay? Because it has too much potential, that's the whole point.
So how do you obtain the adult stem cells from a baby?
Well, okay, so first you need consent from the parents, assuming that this has some kind of benefit either for the baby himself or for a sibling or for society at large, you know, for the advancement of medicine. So once we have consent, and the consent is going to be predicated on the fact that there's no significant risk or harm to the baby. So you can use the bone marrow, you can use the baby teeth when they're shed. There's no danger or injury to the child when we shed our baby teeth, right? Here's the baby tooth. You put it under the pillow or you take it to the lab. You see? So that's how we obtain them. Mm-hmm. From adult, it's easier because we can have informed consent directly from the adult. Typically, a patient who is injured or is ill is going to consent if there is a potential for healing his or her illness with their own stem cells. So that's how we obtain them. The more drastic case would be bone marrow. You have to actually go into the bone with a large bore syringe. Typically, they do the os de la pelvis, the pelvic os, the circle here on the pelvic bone. Okay, you can feel it with your hands. But now they can even obtain stem cells from blood. You know, pheresis. I don't know if you've ever done, if you've ever donated blood. Okay, but there's another procedure that you can do, which is called pheresis, which it starts similar to a blood donation, but instead of just taking your blood into a bag, they put it through a machine, and that machine is essentially a centrifuge that will centrifuge out just the platelets and pump back the blood to you. It takes several hours. So done pheresis, yeah.
Yeah, for was cancer,
Yes,
and so I needed platelets.
exactly.
And so they pull it out of one arm,
Yeah,
take out the platelets
out,
and put it back.
and in comes through the other arm,
And
similar
put it
to,
back in the other arm. So
exactly.
45 minutes, you just sit there.
Yeah, yeah, it takes a little longer
Yeah.
than a blood donation,
Yeah.
but it's for the platelets specifically, okay? And in that sense, it's kind of the machinery is similar, the procedure is similar to dialysis, in that they take out your blood, filter it, and pump it back in. But in the process, I get very cold. The body gets very, very cold. So I'm asking for blankets. And by the end of the procedure, about 5 blankets on top of it, because they have to, they cool the blood down for the process. Okay. Excuse me for a moment. I need to pause for a moment. A moment. Okay. Okay, apologize for the pause. We continue now. So I was explaining that for embryonic stem cells in clinical trials of with experimental animals, with mammals, typically they cause tumors, okay? And so they're very, very sparse results that have happened in animals with embryonic stem cells. In fact, this research has been going on for decades now, and to this day, to my knowledge, there's only been one attempt with clinical trials on humans with embryonic stem cells, right? With human embryonic stem cells. And that was a few years ago from California, Axala, no,
Really?
what's the name of the?
California.
Yes,
Yeah.
yes, what is the name of, it was one of the early biotechs in California that started in the 70s. And they wanted to do a clinical trial. They did something that they thought could get some results with macular degeneration or something like that. It was not life-threatening or anything, but it could cause blindness, and trying to regenerate the retina of the eye or some layer of the eye by injecting embryonic stem cells, human embryonic stem cells. Okay, so after many years, they were able to get a protocol approved by FDA, the Food and Drug Administration, which is the government agency in the US that certifies or gives permission, approves clinical trials for experimental, for drugs, for example, for medications, for pharmaceuticals, and also for any therapy, experimental therapies like cancer therapy or things like this. So embryonic stem cells, of course, it's experimental. And so this company was, the biotech was able to obtain the first license from the FDA to do these trials. Well, you know what? They started the trials, very ambitious, very sophisticated, millions of dollars went into it. And after a few months, they had to stop the trials. And they retrieved and they pulled out of that license because it turns out they were having issues with the embryonic stem cells, with human embryonic stem cells being implanted into patients that were having this, I think it was macular degeneration, where they were beginning, there was a possibility that they would develop tumors. Okay, exactly. So these techs, these biotechs, you know, it's a high risk investment. If it works, of course, they will make millions because they will be the first one to pioneer procedures like this that potentially could heal any kind of injury in the body and so forth. But being high risk, they can also lose big time. So they pulled out of the research before they got sued to the, you know, taken to the cleaners with it. And precisely because of the difficulty of the embryonic stem cells, there's just too much potential. And that's the only trial that I know after decades of this research going on in humans. Okay. John, question?
Yeah, coincidentally, in my pen right now is Baskin Palmer Eye Institute from when I visited last year
Yes.
at University of Miami.
Yes.
We met a PhD student who's doing exactly what you're saying.
Yes.
To replace retinal cells, but it was with the dental adult stem cells.
Yeah.
And
okay.
it was
Adults.
successful. It's working.
There you go.
So she's doing her whole thesis on that. And so that's how long it's taken now for her to, I mean, I'm assuming she's one of the first people around to be doing that.
Yes.
So they're curing blindness with it, I mean.
Yes, exactly.
But is the federal government still funding embryonic stem cell follow?
Yes.
I know,
We'll go
I know
there.
the last administration
Yes.
was the one who granted money.
Yes.
Actually split the money of stem cell
Yes.
research and put some into embryonic stem cell. That's still going on?
Yes, I'll get to that in a few minutes, the politics of it, starting with George W. Bush, moving on to the Obama administration, and now with the Trump administration. But yes, like I mentioned, there's technology, there's economy, and there's politics involved. And then there's also ideology involved here. Okay, I'll get to that one. But I wanted to direct you to this website, stemcellresearch.org. It's all a single word, and I put it in the PowerPoint so you'll receive it. But you just punch that in. And this is a website that has been developed by a number of researchers, clinicians, and theologians that point out the number of clinical trials with adult stem cells, human adult stem cells, typically autologous, that are successful. The last time I checked, which was about a year ago, they had about 70 successes that are documented clinically with peer-reviewed articles in scientific journals. About 70 different types of successes at different levels of clinical trial, but definitely on the positive on success. This is all with adult stem cells, not embryonic, all right? And because they come from people who have consent or parents who have consent on behalf of their children, no one is killed for obtaining adult stem cells. No one is injured significantly, other than a puncture or something like that, to obtain them. So ethically, they're okay. Ethically, they're certainly okay. And like I said, providentially, they're giving results. Precisely because they're multipotent, they're not pluripotent. So they're already toned down in a particular organ system, okay? And the ones that are being most effective in general right now are the ones that they're known as hematopoietic. In other words, they come from the vascular system for healing injuries of the heart and so forth, okay? Because they're already coaxed into the vascular system, the circulatory system, which also associated with muscular system. So we have all these results. Let me see. This is a group that is called Do No Harm. And you can explore this site, but it's a good reference. Here, used to post.
Are there any statistics on embryonic success stories at all?
Not in humans. In animals, yes, a few. A few in animals, like that trial of the rat that I mentioned, all right? In some of the rats, they gain part of the motion of the lower limbs of the body, but typically the lower limbs. So the rat would, arrastrarse, would crawl with the forward limbs. The rat would become paraplegic, in other words, from the chest down. The hind legs, typically the hind legs and the tail, they would drag. And some recovered partially some of the movements. So there's been some partial success. We have to be honest and say it, but it's after going through many rats, and the majority of the rats did not heal, but actually got the tumors. So how to separate them?
So what's the political reasoning behind this whole process if we have like 70 cases, success stories with the adult stem cell research? There is no harm involved, yet the other side of the coin implies harm, and there's zero success. Why the government still funding that route?
Right. At the end of the day, it's about the ideology, and it's also an anthropology. In other words, they obviously don't see the human embryo or the blastocyst as human or as fully human. Okay, they have to not see the human embryo as fully human, because otherwise they would have to admit that they're killing human embryos. They're killing some people to heal some other people, and there's no ethical justification for that. But we'll get to that in a little more detail. I just wanted to follow the sequence here and point out here's their bottom line. To date, they have 73 cures at different stages of development with adult stem cells, with embryonic still zero. at the human level, all right? And you can follow week after week. They publish all the articles that, that are happening. Interesting, I just glanced at this one. During retractions. Retractions are when experiments are done that are iffy and questionable, and then because I'm zealous, here in Kyoto University, Japan, some zealous researcher who has not the scruples that we have will try to publish something, and then sooner or later it's found that it was bogus, it was doctored, it was fake, and so the journal has to retract that article and go in embarrassment and shame and admit that there was some fraud occurring. There's a lot of fraud occurring in science today because of the pressure for getting money for continuing the labs for the research. Okay, so a lot of politics in this. Okay, so let me get a little bit into that. What happens here? Because at some point I go into, oh yes, the iPS cells. Let me see. Okay, so for the sake of completion, let me cover. No, here's a good time to talk about the ethics and the politics. All right, so what do we have? We have that clinically adult stem cells are working. Again, I'm talking about the human, and embryonic stem cells are not working. Why are some researchers and some clinicians intent on continuing the embryonic model? Okay, well, it comes down to money and maintaining the profession, maintaining the research going, their source of income, of professional income for the labs. Because they're saying, okay, so these human embryonic stem cells, these ICMs, they're causing tumors, right? Because they're pluripotent. Well, how about then we use them for cancer research? And this is the segue. Why? Because in a sense, cancer cells are, they're tumoral cells. Why? Because they continue to grow uncontrollably. See? They don't react to the normal control of shutting down once they hit confluence. Confluence is when a cell line or cell layer reaches the edges of the Petri dish or getting too clustered, getting too high density, right? It's called confluence. They stop growing. That's a normal cell, normal tissue, so that we don't develop tumors all over the place. So cells have this signaling that occurs in between them in communication, cell-to-cell communication, where there's enough cells in the tissue, they stop growing. They go into G0, okay, in the cell cycle. Or enough of them die so that enough of them are replaced and you get a net zero growth. Like what happens with RBCs. RBCs, red blood cells, we're producing millions of red blood cells every day because they get wear out. And millions are dying. So what happens to all the dead RBCs, how they recycle and reabsorb? Remember nature's economic? Yeah? Recycle in the body. The spleen filters out from the blood the old RBCs, and the spleen digests them and reabsorbs the material, the organic material, and then is recycled into making new RBCs in the marrow, okay? In the millions every day as we go along, so that there's a net zero increase. So these embryonic stem cells, they continue to grow uncontrollably, so they're actually, biologically, they're good models for studying cancer. And because there are so many different cancers and the incidence of cancer seems to be rising, it's hard to tell if it's just that it's being reported more now and detected more, or the actual incidence of cancer is actually increasing, which then will we speak about our unhealthy lifestyles and the increasing contamination of the environment that is with carcinogenics and so forth. But one way or another, there's more report of cancer today. And so that's a big, that goes into the billions. That's a big multi-billion-dollar industry is cancer research, right? The American Cancer Society, and from there on, the purple ribbon, the pink ribbon coming for the cure and all this, big. For the women, breast cancer is a big killer. For men, is prostate cancer is a big killer. So, you know, a lot of money is being put into that not only in the US, but throughout the world. In what we call the first world, or the industrialized or the advanced societies, billions of dollars are put into that research. So how about we develop a human model of cells that behave like cancerous cells, and we can research with them. And those are what the human embryonic stem cells are becoming. And so just to connect, to link one atrocity with another, with the embryonic stem cells and these scientists, unscrupulous scientists that are intent on experimenting with these human embryos, link it with the IVF industry, in vitro fertilization clinics, and the 1 million human embryos are frozen, cryopreserved, that the clinics don't know what to do with them because the parents already walked away with the child that they wanted. All right, now the IVF clinics can donate those embryos to the labs, and I put donation in quotations because, sure, they cannot sell the embryos because it's illegal to sell human embryos, but they donate them. But that clinic, the IVF clinic, is entitled to compensation, to monetary compensation for having developed those embryos, for having preserved them for so many years, et cetera, et cetera. So in come the thousands and thousands of dollars that they can charge, and they set the price, right? Because they're the clinic with the markup and everything else. They set the price of actually existentially what those human embryos are worth. So you see how the circle is completed? IVF clinic is feeding embryonic stem cell research in cancer research labs in the US and throughout the world. And to add insult to injury, the big bucks come from NIH, National Institutes of Health, which does the human research, and NSF, National Science Foundation for the non-human research, billions of our tax dollars, right?
Father, not that this is possible, but what if this happens where they don't have any more embryonic stem cells? We run out for whatever reason.
Exactly.
We burn out the 1 million that we have in
Right.
cold storage.
They all get researched out. Right.
Now we don't find out cure for cancer.
Right.
What do we do now?
Okay.
Where do we go for that?
Yes. You know where we go? We go into hybrids. This is actually the last lecture of this beginning of life course in a few weeks. All right. Hybrids. In other words, developing substantially human sexual organs in animals. Yes, you want to drop your jaw? It sounds like science fiction, right? Something that you will see Saturday night at a science fiction movie. Well, this was actually proposed by NIH, our NIH in the United States, the National Institutes of Health. This memo circulated through the American population back like in September or so of last year, just before the latest presidential elections of November, that the NIH, currently, there is a ban on growing substantially human organs, especially a brain or sexual organs, in an animal, a lab animal, like a mouse or a rat, okay? There's currently a ban, thanks be to God. Well, the NIH, in September of last year, asked the American population if that ban could be relaxed enough to grow. These are the actual words, and I'll send you the text when we get to that lecture in a few weeks. Substantially human brains in a lab animal. That was one proposal, and a separate proposal, substantially human sexual organs, again, in an animal. In other words, they're looking for an infinite source of human eggs. In other words, growing an ovary, human ovary in a rat. Bottom line. So you have an unlimited supply of human eggs. Or growing a human brain in a rat because of the Human Brain Project, which covers every aspect of the human brain that one can imagine. Very ambitious, very unethical, put out to the general population just in case the voting didn't go according to that ideology. Okay, thanks be to God. Some of us picked up the memo, the email, and we started, I spent about 2 days, almost around the clock, sending emails out to hundreds and hundreds of people to vote against this. Well, it turns out that they put it out for consultation because they have to, they ask the American public, it's public money. Thanks be to God, the response was so overwhelmingly no to the NIH that the NIH for now has desisted. It's stopped for now.
Gonna come back again.
Absolutely, it's the wave on the shore, right? This wave passed, the next wave will come again. And it's only a question of administrations.
Put another ideology into the government and it comes right back again.
Absolutely. Because again, there is the potential, if we just look at it ideologically, the ideology, of course, is just the utilitarian ideology of pragmatic of what works. Why do I have to bother with hyper-ovulating a woman, which they admit is risky and kill the woman and have to pay her thousands of dollars for her eggs if I can just do it on a rat, okay? Obtaining human eggs. You know what I'm saying? And the same with the brain. Why do I have to go for consent on a human brain experimentation, a trial that is very difficult and has all kinds of restriction on the protocol on a live human being if I can do a human brain in a rat and experiment to infinity? But that's the problem is with a pragmatic utilitarian mentality. If it works, do it. That mentality, when you take it to its limit, it goes into the absurd. It goes to the absolutely absurd because there are no limits. It turns out that the end justifies the means. Of course, no one questions the good ends. The good end is to cure cancer, is to cure all the neurological problems, and to eventually get the human that lives forever somehow here on Earth. Who wants that? Anyway, but that's the end. Laudable, very beneficial, etc., etc. But the means also have to be justified. And this whole program, not just this course, but the whole program is about justifying the means. John.
I guess the only benefit you're talking about is, like you mentioned, is studying the embryonic stem cells and watching how when they get disease and how the disease progresses. So
exactly. Exactly. Okay. So for that, the purpose, the ends are good in themselves. They do want to study cancer progression because cancer is a very complicated system. And it depends really on what organ, where it occurs in the organ. Some cancers are very aggressive. Some, we have all kinds of skin cancer that take 200-300 years to develop. So who cares?
My mother is putting this stuff on her nose and her face right now.
Yeah.
It's eating away at her skin.
It does. Exactly.
Something or other, and it's to get rid of the cancer, and she has to keep doing it, for weeks.
Exactly. It's a little tube like this. And you put just a little bit of that ointment, okay, topical, and that little tube costs about $3,000, $3,000. Yeah.
Is in the medical field.
Thanks be to God, there's insurance. Okay, but yeah, what it does is it accelerates tremendously mitosis. So the cells rapid growth, and then they destroy each other, and so it's peeling away at the skin layers. And again, thank God for stem cells, because then the new skin will come out, and that's what it's doing. It's killing all the good and the bad skin cells, and then new skin will come out. Mm-hmm. But many skin cancers, they take 200-300 years to grow, so who cares, all right? But other cancers, in a week, spleen cancer, fulminating, it can literally kill the whole spleen in a week, and we can die. So a lot of research into cancer, into cancers, and therefore big money and big pressure to publish, to be the first and discover a little protocol and so forth, and to get more research money. And the big bucks come from the federal agencies, which is public money. Okay, and that's why it becomes political, and that's why it's important to vote and to know about the issues. Okay, to complete the picture, we have to look at IPS. Okay. IPS are induced pluripotent stem cells, IPSs. IPSs. This guy is Yamanaka. It turns out that Yamanaka was a scientist in Tokyo, in Japan, and he was doing embryonic stem cell research. And he was looking at those blastocysts. And he was reflecting, I don't think he was even a Christian at that time, but he was reflecting as he was looking at the blastocysts under the microscope. He had enough honesty and integrity to say to himself, these blastocysts look very similar to what my daughters looked like when they were at that stage of development. Because he published this in an interview that he did. He says, there's got to be an alternative that is ethical. Because he realized that by dismembering human blastocysts, he was killing those human embryos. Because those are human embryos, just like his daughters were at that stage, and himself was at that stage, and every other human had been at that stage when we were blastocysts, in effect. All right? So he says, there's got to be another way. And what he thought was of undifferentiating cells, de-differentiating cells, going backwards, okay? In other words, he was talking about undifferentiating, fully differentiated cells to try to reverse the process of nature. Remember that I told you that in nature, differentiation, cellular differentiation is a one-way street, all the way from totipotent originally, and the zygote, to fully differentiated in the trillions of cells that form our bodies, right? Okay, so that's a one-way street differentiation as growth and development occurs. That's precisely what is known as development, organically, biologically speaking. We said, what if we try to reverse that process to undifferentiate differentiated cells? And that's how it worked on. And essentially it comes down, it boils down to about 3 or 4 factors that are developmental factors. They control development early on in the embryonic stage. And he actually discovered what these factors, when you hear the word factor, typically we're talking about proteins. These are regulatory proteins early on in the embryonic development, precisely at the blastocyst and the gastrula stage, when the cells are being differentiated. That is regulated by about 4 of these proteins that are regulatory proteins. And when he was able to arrest those proteins, when he was able to inutilizar, to block those proteins, the cells would differentiate, will undifferentiate, will revert back to a previous potency. So he got cells that were multipotent to go back to pluripotent. But he did not take them all the way back to totipotent, because if you do this procedure backward enough, you end up back with a morula or a zygote. And you're back at potentially generating a zygote, okay, from a fully differentiated cell or partially differentiated. So bottom line is he went from multipotent or fully differentiated cells like fibroblasts, all right, into pluripotent, and was able to switch cell lines from muscular cell lines to, I don't know what other, maybe neurological cell lines. But he went from multipotent to pluripotent backwards, okay? Something that normally doesn't happen in nature, but because nature is so mechanical in its processes, the proteins in charge of that development were identified and blocked out, so the process reversed. And that's why they're called induced. They're called induced because this is done in the lab artificially by blocking these proteins. And they are pluripotent, okay? So the acronym is IPS, small I, for induced, IPS cells, okay? So you'll see in the news, IPS, it was a big hit. I think he got the Nobel Prize for it. And this can work, but there's still some issues with it. It's not fully perfected. But the bottom line is that there is now an alternative to try to get pluripotent cells. So this could work for cancer research, for example. It's a healthy alternative, it's an ethical alternative to embryonic stem cells, and obtain pluripotent cells for cancer research without having to kill human embryos, okay, by using IPS cells. And these are the IPs. I should put here, yeah, Maraca somewhere.
So in that case, with pluripotent cells will have the same outcome than totipotent cells when it comes to cancer research, because they can argue, well, yeah, but it's not as good as a totipotent cell, so we're still not there yet. Right.
Well, the totipotent is really for cancer. You don't want totipotent. You want pluripotent.
Oh,
yeah, you want pluripotent. Okay. And so this could do the trick. The argument is that, well, they're not natural. They don't come from, this is an artificial procedure done in the lab. So, exactly. So there's always an argument, okay? Of course, the bottom line argument from the utilitarian camp is that they just don't see the human embryo, the early human embryo, as fully human. They fall into what is known as proportionalism. All right, proportionalism is a theory or a worldview, a concept to see things in proportion. And certainly the human embryo is growing, right? There's no doubt that a zygote doesn't look like a gastrula, and a gastrula doesn't look like a fetus, and a fetus doesn't look like a baby, and a baby doesn't look like a teenager, and a teenager doesn't look totally like an adult. So there are stages, and therefore they say, well, because this tissue, this thing doesn't look fully human, then it is not fully human. And they confuse stages of development with an ontological status, what we call an ontological status, this is philosophy, a status of being, okay? If the human embryo is not fully human, at what stage is that embryo fully human? Back to previous classes, the argument is that any stage after the zygote is arbitrary. Is it going to be the heartbeat? Why the heartbeat? Is it going to be the neurological development? Why neurological? You know, is it going to be birth? Why birth? All those other steps are temporary, are partial, and are arbitrary. The only one that is qualitatively different from the egg and the sperm is the zygote, the beginning of the new individual. And so we have to grant that zygote full humanity. Of course, with further stage of development, but full humanity. See? And that's where philosophically they just don't get it. And in my opinion, with all due respect, and probably unconsciously, they're all just lazy. They don't want to think about it philosophically. They don't want to go deep enough with a philosophical argument of what we call an argument of logical consistency. If you say that a child is as human as an adult, what's your basis for that? A child doesn't really look like an adult, not totally. A child, by definition, is not sexually adult, is not reproducible. It does not have sexual maturity. So why don't you put, really, a child cannot reproduce, cannot procreate by him or herself. They have to wait at least another decade or so to be sexually adult mature. So why don't you put humanity, really the capacity to generate a new individual, why don't you put it on the adults? We're not fully human until we become adults. Then by that logic, we could kill, we could experiment without their consent, anyone who is not an adult. And so they're illogical, really. And these people are very intelligent. These scientists are very intelligent. That's what I make a distinction between intelligence and wisdom. Okay? Because I know some people who are very intelligent, but they're not wise. And I know some people who are very wise, but they're not that intelligent. But they have a wisdom about life, and they have a reverence and respect for life, and they ponder things, and they don't have PhDs or anything of the sort. I know a good number of PhDs been in this world for 30 years now that are very intelligent, bright, but not necessarily wise.
Well, the 7 gifts of the Spirit, Father.
Yes, indeed.
And they're 2 different gifts.
That's right. They are 2 separate different gifts. And they are for our benefit.
That's right.
So that's my little speak about the biology of stem cells, the technology of stem cells, the economy of stem cells, and the politics of stem cells. Okay. So you see how this is a complex world, and we're trying to present the arguments of logical reasoning based on the facts of nature. To finish the stem cell, and we'll take a break before cloning, is that third option also that I talked about, actually fourth option. So we got embryonic stem cells, we got adult stem cells. Now we got IPS, induced pluripotent. The fourth option are umbilical cord stem cells. Let's look at umbilical cord stem cells. Umbilical cord stem cells come from the umbilical cord. But it's not the cord itself, it's the blood inside the cord. Okay? Now what's the umbilical cord? The umbilical cord are the tubes or the blood vessels, veins and arteries between the fetus and the mother and the placenta. Okay, that's the umbilical cord. We know that it keeps that fetus alive during the whole time of pregnancy. Once the baby is born, the placenta is also born, if you will. Okay, the placenta has to be expelled from the mother's uterus, and connecting the baby to the placenta is the umbilical cord, which is mostly the arteries and veins. That has to be severed, cut, because the placenta has finished its function. Notice that the placenta, by the way, is a vital organ, keeping the unborn alive, but it's a temporary organ that is shed, discarded at birth. But it's a vital organ belonging to the baby, okay, to the embryo. Anyway, the umbilical cord then is severed, and the little dot tied and pushed into the belly, and that becomes our belly button, our umbilicus. All right, there is blood inside the umbilical cord. That blood belongs to what? The mother or the baby, organically, biologically, from the baby, right? Even though there's passage, their actual research has shown that cells from the baby go into the mother's circulatory system, and cells from the mother go into the baby's circulatory system through the placenta. Okay, a few, but they do. In fact, the baby is capable of messing around and changing the mom's immune system and vice versa. So there's a lot of interaction going on there, but the cells that are inside that blood, that cord blood, are predominantly from the baby that was just a fetus a minute ago before birth. And so these cells have embryonic quality. In other words, these cells, which are mostly, like I say, hematopoietic, or they're cells that are predisposed for the circulatory system, all right, for the vascular system, these cells have embryonic quality because they're pristine, they're young, they have not been exposed to ultraviolet radiation or other chemical toxins and so forth. So they are not supposed to have too many mutations in them, et cetera, et cetera. All right, so they are kind of embryonic quality. However, normally at birth, the umbilical cord is discarded, thrown into the garbage, biohazard, considered medical waste. And typically it's incinerated. So they're thrown away, okay? Because they're not even taken out of the tube, out of the cord, out of the umbilical cord, it's inside. Well, these umbilical cord blood cells, which some of them are stem cells, they can be saved now. They can be saved, okay? And these are umbilical cord stem cells, otherwise known as cord cells, okay, or cord blood. It's not the actual blood that is saved. The blood is first retrieved after the baby. Follow the sequence. The baby is born, the tube is tied, cut, the umbilicus, tape on the belly of the baby. Now the baby is away with mom, feeding, whatever. Now I got the cord, or the cord is, the blood is extracted. Actually, I think it's extracted once they do the knot, but before they cut it. So the blood is extracted to keep it in a sterile environment. Now the cord blood is in a syringe. Baby is fine and safe with mom, all right, no problem. That cord blood then is spun in the lab and separated to separate out the stem cells. And then those stem cells are what? Cryopreserved, dipped in nickel nitrogen, 180 degrees centigrade. They crystallize and they're saved for baby because they're immunocompatible, right? They come from the baby, would be an autologous transplant. Hopefully they'll never be used. Okay, hopefully that baby will grow to an adult and die as an old man or woman in their 80s, live the full life, no injuries, no diseases. But if something does happen along the way, decades later, one could go back to that bank, and hopefully some of those stem cells that were frozen and crystallized will serve and have their potency. Okay, there's a good chance that they will. For example, there was in the news maybe last month of a 25-year-old girl who was impregnated with an embryo that had been cryopreserved for 25 years, precisely. Okay, and the baby, and now it's published. They waited several months to see if the baby was going to live and all that, and now it's been published. So that was a success. We don't know about how many trials were done or anything like that, but just to say that at least for that baby, for that child survived 25 years in suspended animation, crystallized, all right? And so it's amazing. It raises a lot of issues. Anyway, so that's the issue with cord blood, okay, or cord blood cells, which are in their stem cells. They're not pluripotent, they're multipotent, okay, because they don't come from a blastocyst, they come from a fetus. So they're multipotent, but they have a lot of potential for healing. And they're ethical, the baby is not harmed, there is consent from the parents, but there is no insurance to pay for it yet. And therefore, the parents have to pay, okay? And there's a financial consideration. The procedure, typically, you can buy the kit, you order it online, and the kit comes, and then you alert with your pregnant wife or and the spouse. They talk about this, of course, beforehand. They alert the doctor and the nurse that they want to save the cord blood. They bring the kit to labor, and when all that happens, it's stored. And I think the procedure, they're charging maybe $1,000 or $2,000 or $3,000, depending on the hospital. It's a huge markup, because how much does it take to draw that blood, right? Humongous markup, quality control, all right. And then, yeah, exactly. To preserve then there's either a monthly or an annual fee, $100, $200, $300, so forth, to the cryopreservation bank.
I think you can use these stem cells on the simplets
Yes,
as well.
Exactly. So then from
that's obviously percentage
of compatibility.
Yeah.
Exactly. So there you branch away from the donor, okay? And as you branch away from the donor, the immunocompatibility decreases, but there's still a good chance. So it could be used for siblings, it could be used even for parents, all right, and so forth. And beyond siblings, it could be used maybe for cousins or any other relative. There would be some percentage of compatibility, but only God knows what actual percentage will be experimentally, okay? So trials have to be done to see if what is the immunocompatibility. And just thinking out loud here, the process, I presume that they would do karyotyping, or they would do some biochemical analysis on some of those stem cells to see what kind of compatibility they have. Minimally, the blood type, well, which would be known from the baby also, and the Rh factor, Rh positive, Rh negative, you know, that's the minimum compatibility, which is relatively easy to detect today. And then more tests could be done to get more sophisticated. Questions, comments, John?
Is there a benefit to the cord cells versus like bone marrow multipotent cells?
Yes, okay, so think about it clinically. Does it harm the baby to obtain these cord bloods? Nothing at all, because that's going to be discarded, okay? To obtain bone marrow, minimally you have to go with a large bore syringe into a bone and extract it. But you can say, well, how about just blood from the circulatory system that we talked about, that just like pheresis, right? Platelets can be obtained. Also, stem cells can be obtained just for blood. Well, you still have to do a procedure on the baby, the child, the adult, whatever, to take out that blood, filter it, and pump it back in. So that's more costly, more risky. So these are still cheaper just by the procedure. The unfortunate thing is that not covered by research, not covered by insurance yet. And what I want to propose is for couples, especially young couples that are undergoing this procedure, is to petition their insurance company to cover it at some point at least partially, start partially. Why? It behooves insurance companies to cover this. Why? Well, if you think about it, as long as there's ethical integrity in the insurance company, it's more cost effective for an insurance company to seek a cure with these stem cells than to cure, let's say, a catastrophic disease. Let's say that potentially this could cure some cancer. Well, it's much more costly for an insurance company to cover the cost of a cancer treatment than to cover a stem cell treatment from cord blood cells, right? So I'm thinking that it has to, for the insurance company, it has to make financial sense. Otherwise, you know, it's exposure. So if enough couples do this and they petition their insurance companies to cover it, the idea is that eventually there will be a critical mass of cord blood banks. And that's the other thing. Now these banks have been nationalized, and the registry, there's a cord blood registry now, national, with the information being shared throughout the nation, so that maybe one couple is saving their cord blood that may help another couple in some other state because there's an immunocompatibility there. And there's no parentage, but there's an immunocompatibility. And so there's a benefit over there. They would pay maybe for some of the storage, etc. You know, something has to be worked out. But I'm thinking that if enough couples save their cord blood at birth, eventually the insurance companies will get it that it behooves them to pay for the procedure, or at least part of it, to partner in. Because in the long run, the more cord blood that we have in these banks throughout the nation, the more potential cures that are out there for less money, okay? And more successful. So that's my thinking, but it's up in the air right now. To my knowledge, I don't know of any insurance company, unless you have a very deluxe Cadillac type of insurance that will cover cord blood recovery and storage. Okay. Okay. So, if there are no more questions about stem cells, just to round it up, we have 4 different types, really. 4 types of stem cells, right? Which are ESCs. What is ESC? Embryonic stem cells. We're talking about the human, all right? Then we have ASCs, adult stem cells. And then we have IPS, induced, pluripotent, and we have cord blood cells, right? And of those 4, the only one that is ethically offensive from our perspective is going to be ESCs, embryonic stem cells. Okay, embryonic stem cells in the human, because a human embryo has to be killed, right? Again, if we were able to obtain cells from the ICM without killing that embryo, without significantly harming the embryo, that's the problem. How do you not harm that embryo? Okay, to obtain them, you have to engage with in vitro fertilization, because you're not going to go into women's womb and pull out a blastomere from a blastocyst that has implanted. Imagine the high risk on that, you're going to cause an abortion. All right, so you have to partner with the in vitro industry, which is intrinsically evil. So there's really, realistically, there's no way of obtaining ethical embryonic stem cells today, at least with the level of technology. All right, but the other 3 are ethical, and the other 3 are actually giving results. So that's where we would love the science to go. I'm going to stop here and take a break for a while, then we'll come back to regroup for the human cloning, okay? So let's stop here. Okay, I'm going to resume recording now, the second part. Having looked at stem cell research in some level of detail, we're going to move now into cloning in general, and then human cloning specifically. Okay? We had talked about cloning a little bit before as a type of what kind of reproduction? Asexual reproduction, right? Because in cloning, there is no fertilization. There are no gametes, there's no egg, no sperm, no fertilization, no zygote. We're talking about an identical copy, a clone of one cell to make another cell. Okay, now this happens in nature. So really all the different types of asexual reproduction, in a sense, is cloning, whether it's binary fission or fragmentation and regeneration or vegetative propagation. What are some of the others? Budding off, mitosis. All these are types of cloning because the progeny, the offspring, the result is identical, essentially identical. I'm talking about the DNA to the parent cell, all right? So diploidy, for example, is maintained from 2N to 2N, right? Chromosome pairs, which is the standard, the normal number of chromosomes in all the species. I'm going to focus on human cloning, which biologically is not different from another animal cloning, certainly mammalian cloning. Biologically, the technique and in the lab is the same. But I'm going to focus on human cloning itself. So again, human cloning is a type of asexual reproduction. It's not natural because we don't know that it happens in nature, except for monozygotic twinning that could be seen as a type of cloning, all right? Excuse me, and we covered monozygotic twinning a while back. Again, making distinctions, we can talk about two types of human cloning: reproductive cloning or therapeutic cloning. Now, these two adjectives are actually pointing to the functionality or to the ultimate end of why the cloning is being done. Why the cloning is being done. Is the cloning being done for reproductive purposes, or is the cloning being done for therapeutic purposes? What are we talking about? Okay, I'm going to get into it, but before that, I need to explain a little bit the cloning technique, just in general, and specifically in mammals. Anyone remember Dolly?
Yeah.
Not the singer, but the sheep, right? So there's Dolly Parton, and then there's Dolly the sheep. Okay, Dolly the sheep was manufactured, was the first cloned mammal. And I have the dates here, 1996. I was in Scotland, the Roslin Institute, which is a big biotech institute associated to the University of Edinburgh, I think, in Scotland. And the scientist there was Wilmot, Ian Wilmot, and there he is with Dolly, okay? Now, and I think this Dolly is already the dead Dolly. This may be the preserved, they killed Dolly and then they had to euthanize Dolly, I'll tell you why. And then they, what do you call that? They made a...
Taxidermy.
Taxidermy, yes, yes. Okay, but that's the end of the story. Let's look at the beginning of the story. Dolly is a clone. Dolly comes from a cell, a donor nucleus. All right, so here we have a lamb who happens to be a female because she has an udder. What is the udder? The udder is the mammary gland of mammals, okay? And they took a cell from the udder. It's significant for a reason I'll tell you in a minute. And they took another sheep, female sheep, and took an egg from that sheep, an ovum. All right, so we have two females here, right? Okay, they're trying to match up these two females as closely as possible. First of all, let's start actually with the egg is better. They take out from a mature female sheep, when she ovulates, okay, they take out an egg, extract an egg, and now they have a mature egg in the lab, mature, yeah, they did. They have an egg in the lab, a mature egg from a female sheep. Now that egg is diploid or haploid? It's an ovum. Haploid, right? It's a haploid, it's a gamete, it's a female gamete. Now they take a cell from a lamb, a ewe. A ewe is a female sheep young, okay? They wanted young because they wanted to have cells that were relatively young. So why did they use a lamb instead of a full-grown sheep? Well, this full-grown sheep is more exposed to radiation, for example, over the years. So there could be some mutations on that DNA. They're trying to find cells that are as pristine as possible. But they're using somatic cells. What does somatic cell mean? Somatic cell is anywhere in the body, soma, body, all right? Somatic cell is any cell in the body that is not a gamete, that is not an egg or a sperm. So essentially, it's every one of our cells, the trillions of cells that we have, are somatic cells, except for egg or sperm, which are gametes. So what is the ploidy? What is the ploidy of our somatic cells? Are they diploid or haploid? Diploid. They have chromosome pairs, right? So every cell of our body has chromosome pairs, the 23 pairs, except for the gametes, which are unpaired. Okay, so this is a somatic cell, meaning it's a diploid cell. The nucleus, specifically, is a diploid nucleus. It has the full complement of chromosomes that make up a sheep, okay? It's a female, they use a female to match up because they're going to do a nuclear swap, essentially. They're going to do a nuclear swap. They're going to change this haploid nucleus of the egg for a diploid nucleus of the somatic cell, okay? So they want it to be a female, that's easy enough to obtain, and they also want this female, the donor cell, to be young so that it's less exposed to radiation and chemicals and other toxins that may have caused some mutation in the DNA. That's the main reason for it. Now comes the actual cloning technique. What is the cloning? It involves an enucleation and renucleation. In other words, this egg, what makes it an ovum? What makes it an ovum? What makes it a gamete? What makes it haploid? The fact that the nucleus is haploid, that the nucleus is a pronucleus, okay? So what they did is they took out this nucleus, extract the haploid nucleus, and insert the diploid nucleus, okay? Insert the diploid nucleus. In fact, what they do is even easier than that. They basically, first they take out the haploid nucleus, and then they fuse these two cells. They fuse the diploid cell, the somatic cell, into the enucleated egg. So what are you using from the enucleated egg? Well, you're using the cytoplasm of that enucleated egg, which is similar to a zygote to an enucleated zygote. In other words, where is the cytoplasm? The cytoplasm of the zygote, where does that cytoplasm come from? The female, the mother, the egg, right? So its cytoplasm, its egg is ovum cytoplasm, okay? And so basically what they're doing is they are generating a zygote without fertilization. Without fertilization. You follow? This is the key. This step here is the key. Two steps. First, they take out the pronucleus of the egg. That's called enucleating the cell. They enucleate the ovum, right? That can be done in the lab under microscopic conditions, sterile conditions with, as you can see here, syringes. This is an egg that is being, actually here's the technique, all right? Here's an egg that is being held in place by this tube. This tube has a hole here and it's a vacuum. This tube is a vacuum. It's creating a little bit of suction here. You can see how the membrane dips in a little bit there. See how it dips in? That creates a little bit of suction, enough to hold the egg in place. Now imagine the egg is 0.1 millimeter, right? Microscopic. So all this is super microscopic, but can be seen because it's a photograph. This is what's going on. So this end over here is a vacuum that is holding that egg in place. Imagine the sophistication of this equipment here to generate that kind of a vacuum that can be regulated precisely to hold an egg in place and to not suck in that whole membrane, okay? All right, while the egg is being held in place, here comes a syringe that is very small. It's actually a glass capillary that you cannot see the tip, but the tip is there because the tip is microscopic, and it's a glass capillary that is hollow on the inside. When I was at FIU, we used to do these, not the cloning technique, but we used to generate these capillaries because they're used as probes, electrode probes. So I was working with aplysia. You know aplysia? Aplysia is also known as the sea hare. And hare as in the cousin of the rabbit. And they happen to have, you see this around? It's a mollusk, all right? It's related to the octopus, to the squid, to the clams, and all those guys that are mollusks. And they walk around on the seafloor and they eat the seagrass that is on Biscayne Bay. These Aplysia, they also put out this purple ink, just like many mollusks do, like octopus and squid. But they happen to have these giant neurons in them that have long axons that can be seen under a light microscope, the actual axon of the neuron, okay? And when I was at FIU in my 70s, I was doing research in not my 70s, these 70s, my 20s. I was doing research with a professor there, and I have to put inside that neuron an electrode that to transmit a tiny little electrical current to detect what is known as the action potential, what fires the neuron signal. And so I would develop these little probes to fill them with a salt to transmit electricity. And this is how you make one of these little probes. It's relatively easy, actually, and fascinating. You take a glass tube that is hollow, all right? And with a burner, Bunsen burner, you heat up that tube, and as you're heating it, you're turning it to get an even distribution of the heat until the tube turns red hot. You can see it literally starting to glow red hot. At that point, glass liquefies. It's becoming a molten liquid similar to the red hot magma that comes out of the ground in a volcano. So when it's red hot like that, you just pull it fast, all right? And as you pull it fast, it will break, in quotations, it would break into two pieces, okay? So now I have two pieces of glass tube that is hollow, and the tube will continue to narrow until you cannot see it anymore with the eye. But if you put that glass tube tip under the microscope, you'll see that the tip will have an end. And in fact, it's funny because you can take the tube and you can follow the tube with your eye until you cannot see the tube anymore because it's so thin, but you can actually touch and feel the tip over here where you cannot see it. So it feels like you're touching in thin air, you know, but you're touching the tip that is microscopic, but the finger can detect the tip. Huh? So it looks like it's, but what's interesting about that tube at the very tip is that it doesn't seal. That little hole remains through the tip. So in effect, you have a microscopic needle, and that's what this is. This is a needle, you know, that is a glass capillary that will allow that is the thickness of one cell. And sometimes you can get it smaller even to the thickness of a nucleus. And here it is. You see the nucleus? This is the nucleus being pumped into an enucleated egg. So this here is an egg, an ovum, that has no nucleus, has been enucleated. And here is the somatic nucleus that is being pumped into this enucleated egg. When we do that, we have in fact generated a zygote, because this egg now with a somatic nucleus, with a 2N nucleus, becomes a diploid cell and becomes equivalent to a zygote, okay? Without fertilization, Okay, so this is what Wilmot and his associates did. They did it even simpler by fusing that entire somatic cell into the egg. The point was to get that somatic nucleus in there. And then allow that somatic nucleus, that somatic cell now, the zygote, to begin differentiation into morula, blastocyst. The blastocyst then was implanted into, they used a third sheep. They didn't use this mother with a, because they wanted to push the envelope in this case and to see if this embryo would then implant into a different sheep from where the egg came from, okay? So this other sheep is not the same sheep. And they tried and they tried and they tried until finally some embryos implanted, but then were discarded along the pregnancy, did not sustain. But finally, one embryo implanted and lasted long enough so that Dolly was born. I don't know how many months later, I don't know how long it takes for a
Now
sheep.
it becomes like the U on top.
Yes, so this is an identical clone of this one. Okay, because the somatic nucleus is what defines the DNA of this embryo. And this sheep was just the donor of the cytoplasm. This other sheep was the incubator. But this little U is the clone of this other U.
The nucleus is that's where everything hides. The nucleus is the most important.
Yes, why? Where is the what is in the nucleus?
Yeah.
The DNA, the genome.
So up to fused cell to that stage, we can say that there's life there. From that stage on, if it would have been human, the soul would have been granted in that stage.
Here, if this was meant to be a human in the mind of God, then God would, would be forced to grant this human a soul. Again, because there's nothing more cruel than God could do that to leave a soul, to leave a human being soulless. So what in vitro does essentially, it forces God to grant a soul. But it was not wanted by God. It is allowed by God, but not wanted. Okay? All right, but so let's follow a little bit more closely what has happened here. See, this nucleus is a somatic nucleus. But when this nucleus came from this somatic cell, which I told you was a cell from a uterus, the udder was a skin cell of the mammary gland of this U over here, okay? So this cell, being a skin cell, is fully differentiated. And when the nucleus went inside the cytoplasm of an egg, this differentiated nucleus reverted, undifferentiated, all the way down to pluripotency. Because by definition, the zygote and the morula are pluripotent. Exactly. This was Yamanaka's thing, but done artificially because this doesn't happen in nature. But it turns out that there are factors in the zygote, in the human egg, in the cytoplasm of the egg. There are these factors that are capable of undifferentiating the DNA, essentially opening up the DNA for full expression. Okay? So this is proof of principle, but the furthest proof of principle was to get Dolly to adulthood and to mate and to produce fertile offspring. That's the final proof of principle, is that Dolly was fertile. Okay? In other words, it was not generating an offspring of the same species, but that offspring to be fertile. Okay? All right. A few more details. It turns out that, I'm showing this very nicely in this diagram, but it actually took about 258 trials to get the one Dolly. Okay? So after, in other words, they did this procedure 257 times before they got one to finally be born. And since then, I can tell you, moving forward a little bit, for mammals, since then, mice have been cloned, and rats, and cats, and in fact, the first cat they called copycat. They all have different names. Horses, cattle have been cloned. Like I said, the monkey has been cloned by China just 3 weeks ago. The dog was very hard to clone. I think it was, no, it was Wusok. Wusok was the other one who was faking human cloning, also in Japan. No, in Korea, South Korea, that fell from grace. He faked human cloning in, forget if it was in the 90s or the early 2000s, and published 2 articles in Nature that were fake, Photoshop photographs, and finally it was discovered. It was a huge disgrace for Korea. So he went off the clinical trials, of course, and eventually he got the dog to be cloned. The dog was very difficult to clone because the female cycle on the dog is very difficult. It's very hard to obtain a mature egg from a female dog, which technically, biologically is called a bitch, a female dog, to produce to detect when a female dog produces a mature egg is very difficult. And that's why when you see, I lived in Mexico City for a while and I watched as a teenager, the whole flock of, whole slew of male dogs chasing a female dog for days. They would be on her trail to detect until she finally would ovulate. It's a drastic scene to see that in the streets.
Question I know I know they do this for a while, like artificial insemination for animals.
Yes.
But when you look at the issues that it might present, like in horse racing.
Yes.
Horse racing, usually a successful racehorse is come from a long line of winners.
Oh, exactly.
How would they control this?
Yes, they do, to the point that as you do that inbreeding, okay, and it's called artificial selection essentially, and we talked a little bit about that last semester with Darwin. Artificial selection, you're selecting for particular quality. In the case of horse races, you're selecting for fast speed but small weight, low weight at high speed, so the tone of the muscle and the skeletal structure of the horse and all that goes into play. You keep selecting for those qualities, those particular qualities. It's called inbreeding, but at some point that horse is becoming weaker and weaker in other parts of the body, and typically, I understand for horses, they become weaker on their ankles. So the problem, the downfall, exactly, the downfall of the horses, the race horses, the ankle becomes weaker and weaker until the point that if the ankle breaks, they have to kill the horse because that horse cannot heal enough to run again, okay? And so periodically what they have to do with this inbreeding of race horses, they have to outbreed. In other words, they have to get a wild horse, or they have to get a horse that is not inbred and breed them out and start again to bring in the strong heterogeneous qualities of the heterozygote qualities of the wild type of the wild horse. Okay, so they have to outbreed periodically and then start again the process. It's elaborate and so forth. Cloning bypasses a lot of that, but you don't know what you're getting from this because you're using one particular somatic cell. Of course, genetics is getting more and more sophisticated, so you can also do some quality control on the genetics of that somatic nucleus. Okay. At any rate, it's so what I wanted to say with the cloning of other mammals. Apparently, it takes about, on average, about 250 trials, more or less. That's what I've read over the years, is that it takes about 250 trials in general to clone one mammal. All right? That's what it's been. The last thing to say about cloning and the techniques, so you see how it works, essentially is enucleating an egg and renucleating it with a somatic nucleus. And then hoping that de-differentiation occurs to the point that a new individual is born, okay? And the last detail is this anecdote, the name, which at first blush
there's no sperm there from a male. There's no,
exactly. There's no sperm, there's no fertilization, and that's why it's literally a clone. Dolly is a clone of the ewe where the somatic cell came from, the somatic nucleus. Essentially identical, a copy. Okay. So the last anecdote is about the name itself, which at first blush is funny, but deeper reality is offensive. They just tongue-in-cheek, Wilmot and his colleagues decided to call this ewe Dolly because she came from the cell from the udder of a female sheep, right? From the udder, which is the mammary gland. So it's just tongue-in-cheek about Dolly's mammary glands. Dolly Parton is known for her abundant breasts.
She has had a reduction, so
yes,
she no longer qualifies.
You see, yeah. She has a fantastic voice. You want to see a nice movie, Steel Magnolias? Ever seen that movie, Steel Magnolias? I love it. Great lady, great lady. So this is.
So what happened to Dolly?
Okay, so what happened to Dolly? Thank you. This is the downside. it turns out that this differentiation, de-differentiation, this reverting to the zygote level had its consequences, okay? Because the result, so the cell here, the somatic nucleus is going like backwards, right? To an undifferentiated state and then forward again. Well, that had its consequences. The consequences for Dolly was that she aged prematurely. She started developing, even as a young, started developing arthritis and perhaps some senility. She was banging into things. It was like she was becoming senile prematurely. So this process of reverting this nucleus, of de-differentiating this somatic nucleus, had its negative consequences, which apparently the second time around does it faster. And the animal ages prematurely. And in fact, this is kind of consistent. They found this with other animals also, okay? That they age prematurely. So at some point she was actually in pain from the arthritis. So they euthanized her, they killed her. You know, it's a humane thing to do. And then they preserved her with taxidermy. So I'm thinking that this is actually the model we can look at.
So it was really not really what you call successful then, Bob.
Sorry?
It was not really successful.
Thank you. So it was successful. It was successful for, yeah, that's the model there. That's the taxidermy model, okay? It was successful for getting a fertile adult after 250 trials, okay? But the adult aged prematurely. Okay? Because of that, the scientific community has generally spoken against reproductive cloning in humans. In other words, we should not clone a human for reproductive purposes because that human is an injustice to that future child because that future child could age prematurely. Okay? And so there's a scientific consensus that we should not do reproductive cloning on the human. But therapeutic cloning is a different story altogether, functionally. All right, so let's look at human cloning on the human. Human cloning on the human. Let's look at cloning on the human. You start with a human egg, okay? The ovum. Then you remove the nucleus, that's the enucleation here, right? Then you take a cell, either from the same woman, or from another one, or from a man, but we need a somatic cell, okay? So you fuse that cell with the enucleated egg, and you get the zygote. Then the zygote undergoes embryonic development. You bring the zygote to the blastocyst stage and then implant the zygote. You can implant the zygote into the original donor of the egg, or you can implant the zygote into this other woman that donated the somatic cell, or you can implant the zygote into a third woman that is neither the donor of the egg nor the donor of the somatic nucleus, okay? And give it 9 months and hopefully you get a baby born.
That's similar to the animal.
Yeah, it's identical. Yeah, it's the same thing because we're organically, biologically, we're mammals. So the technique essentially in principle is the same. But what I'm trying to say is that this baby, right, if this was done for reproductive purposes, in other words, for generating a new human being, could have potentially 3 mothers. Can have the mother who donated the egg, can have the other mother donated the somatic nucleus. And the third mother is the one who gestated the pregnancy and no father. So this child could have up to 3 mothers, actually 4 mothers. Who's the 4th mother? The 4th mother is the rich mother who could not get pregnant, who wanted a child and paid for all this procedure. You know what? It's legal. Okay? It's not illegal, so it's legal.
So when it comes to the sex, it's determined by the donors.
Which donor?
It's a clone, right?
Which, yeah, but which donor?
The one that we extract
The egg or the cytoplasm? I'm sorry, the egg or the nucleus? The nucleus, exactly, because the nucleus is the DNA.
So if
The egg is just a cytoplasm.
If it was a man, then the offspring
would be a man.
then
It has to be a man.
that donor could be considered the dad.
Yeah, yeah. So exactly.
So,
but you see, the dad is also the identical twin of that child. So
identical twin, physiologically speaking.
Yeah, DNA is a clone.
DNA, so exactly the same everything.
Right. So that person is simultaneously the father and the brother of that child.
But this doesn't happen now.
It could, because it's not illegal. But what I say is that the scientific community has spoken against this
Because of the premature.
because of the premature aging, the potential premature aging of this child, which happened in other mammals, which has happened in other mammals, okay? But it's not illegal. So anyone who has enough money to pay for it can do it, and anyone who wants to get enough money for donating a skin cell, anyone who wants enough money to donate an egg, and anyone who wants to just take a child for the heck of it and the money of it, could do it.
So it's a level father. This is one way that same-sex couples could.
Yeah. Same-sex couples, single couples, yeah. But they can also do in vitro, which is even less risky than this. It doesn't have to go through so many trials, right?
Less painful.
Yeah. Mm-hmm. Yeah. But you see, so we have essentially, we're trafficking with human tissue, with bartering, with human lives. This is what's going on. This is, sad to say, this is like a pragmatic utilitarian mentality. Okay, if I can pay for it, I can do it. If I can do it, that's the technological imperative. Anything that can be done will be done sooner or later. Forget about the ethics. If I can do it technologically. So it's all about the technological advancement. If it can be done, sooner or later, it's going to be done. That's called technological imperative. Okay? And it's out of the utilitarian mentality of don't worry about the ethics. It's not illegal. So if you're so concerned about it, make it illegal. You know, but until it's legal, get out of my way because I have the money to do it. So that's why I say, because precisely of the potential for this child to age prematurely, the scientific community have really spoken up against reproductive care. It's an injustice.
So
Okay?
science has become one new tower of Babel.
Sorry?
Science has now become one new tower of Babel.
Oh, yes, yes. Yep, sure. Right? Because the technology determines, right? If we don't control technology, technology will control us.
Sorry.
And technology means economic growth. All right. Different is therapeutic cloning. Let's look at therapeutic cloning. So it's all about the purpose. For what purpose is the cloning being done? In reproductive is to generate a new human being. Thank God the scientific community is against it, so you're really not going to find any clinician to do it because it's not good medicine, right? You could generate a child with premature aging. And then the prospect is not nice, which would be to euthanize the child when he or she develops arthritis. Thank God that's illegal. Now, therapeutic cloning, very different. What's the purpose? The purpose is therapeutic. In other words, to try to find cures with this kind of cloning. Well, what's the issue of trying to find cures with this cloning? Precisely to obtain embryonic stem cells. In other words, to generate blastocysts, right? Here we have a zygote, here we have a morula. After the morula, we have the blastocyst, and the blastocyst, any mammalian blastocyst, is going to have a trophectoderm, and it's going to have, is going to have an inner cell mass. So this would be a way of generating embryonic stem cells without the process of fertilization. You still need the human eggs, and you still need a somatic nucleus. We got somatic nucleus all over the place, all right? So basically, the big issue is obtaining human eggs, which can be bought because it's not illegal to give women money for their eggs.
You need the somatic nucleus or the cytoplasm?
No, the cytoplasm, the egg. The egg is the one that's expensive because that needs to come from a woman's uterus, all right? the woman's ovary. But today, you go to colleges, you go to universities, and you look at some of the brochures that are on campus, and they are fertility clinics advertising for donation of eggs with compensation. Okay? And you see them even in airlines. Look at the flight, next time you fly, look at the little magazine on the flight airline, on the flight. There will be some advertisement for compensating women for donating eggs. In other words, undergo hyperovulation and donate us your eggs, just one cycle. Donate in quotations, because they're entitled to recompense, they're entitled to be compensated for the effort. And the effort now, the standard effort is about $10,000, just without any quality control. If the woman is young, then that goes maybe to $20,000. And if the woman is an athletic, then maybe $30,000. And if the woman has a high IQ and can demonstrate it with very good grades, A+ in her grades in college, maybe she gets $40,000. If she's blonde and blue eyes, you know, so it is eugenics. It is quality control. It is selecting, artificial selection on the woman. The younger, the prettier, the more intelligent she is, the more she can get compensated for donating her eggs. This is the industry. This is the absolute truth. You go to campuses and you find the magazines, and you go to airlines and places where people fly and affluent people, and you find the advertisements.
Margaret Sanger all over it.
That's right. That's right. Eugenics. Adolf Hitler, yes, purifying the race. Yes. All right. So therapeutic cloning is essentially trying to obtain embryonic stem cells. There's a diagram. Okay. Patient donates a somatic cell. So, okay, this is the case of the patient. So you can get now the somatic nucleus to come from the patient. So this would help on the issue the patient is generating his or her own embryonic stem cells. So you bypass the immunosuppression, you bypass the immuno incompatibility, right? Because the somatic nucleus is no skin off my nose if I'm a patient, literally, to donate some of my nose skin for the somatic nucleus. Okay? So the patient can donate the nucleus, which would make those embryonic stem cells 100% his or her DNA. There's an advantage. There's no incompatibility, immuno rejection. You get it? So there's that advantage. Of course, there's a minor detail of you need some eggs, enucleated eggs. So you always have to depend on women and the in vitro clinics to sell you some of these eggs. Mm-hmm. With the consent of the woman, of course. Well, we'll do the trial for free for you if you donate your eggs. Mm-hmm. Okay, and the idea is to obtain the cell lines, the 220 cell lines that I say could potentially heal all the diseases and illnesses and injuries that could happen to us, with cell lines that are immunologically compatible. Okay? Big incentive, big tantalizing.
So I don't understand the process. The purpose.
The purpose is therapeutic. The purpose is to generate cell lines that are compatible with the patient.
So the same donor will be for his own benefit?
Yes. The donor of the somatic nucleus.
The somatic nucleus.
Okay? There's, like I say, the detail of the egg, of the cytoplasm of the egg. That comes from the woman, but it's only the cytoplasm. So there's no DNA there's no genetic inheritance. Okay? There's a mitochondrial DNA, no problem. We all have our maternal mitochondria. But the DNA, the genetic information is coming from the donor or the patient who is ill somewhere or has an injury. But now, theoretically, with this protocol, it will be a way of generating tissues and tissues that are compatible with the patient, because the stem cells, the embryonic stem cells, come from his or her somatic nucleus. Okay? So there's that big, big advantage, if you will, the clinical advantage. There's no rejection. And because there's no rejection, in principle, these perhaps should not cause tumors. But because they're embryonic, they're still going to be pluripotent. So they could cause tumors. Okay? they could try to further differentiate these cells, these embryonic stem cells, into multipotent in the lab before implanting. Okay, they could try to get these embryonic stem cells to go multipotent in particular cell lines before implanting to try to avoid the tumor ratio. So there's potential there. There's clinical, there's biology that is compatible. What's the issue? Well, the issue is that once we put a somatic cell into that egg, we are essentially generating a new human being. It's a clone. It's a zygote. Okay? And so an argument of logical consistency would argue that God has to grant a soul to this. If this is meant to be a human being, if this is going to develop into a morula, blastocyst, and implanted would become a human being, even if he or she matures prematurely, ages prematurely, then God would grant a soul. God would not leave the zygote unsouled. All right? And so, in fact, therapeutic cloning is generating human beings in the lab, just like reproductive cloning. It just has a different purpose. In a certain sense, you can say this is twice as offensive. Number one, because it is manufacturing a human being in the lab. And number two, that human being is manufactured specifically to be killed for his or her embryonic stem cells. So it's a double offense. At least in reproductive cloning, you want to have the child. You want to give life to the child. But in this one, the embryo is going to be killed. So
it will never be implanted.
Correct. Because all they're trying to do is obtain the stem cells. It's therapeutic. So it's a double offense.
Bring the stem cells back to
the injured person, to the adult who needs the healing, where the nucleus came from. Okay, so it's compatible. It's compatible with the immune system of the donor, of the patient, but in the process, a human being was manufactured, and then that human being was killed, destroyed for his or her stem cells, for the ICM.
Yeah, I was gonna, real quick, there's a movie about that called The Island.
Yes,
where,
of Dr. Marrow.
Yeah, they all live on this island, and then they start to figure out all these people, and they were told it's a big lie. They start to figure out that they're actually clones from really, really wealthy people in the real world. You know, if they needed an organ.
For spare.
What's the name of the movie?
The Island.
That's The Island, which is a further development of a previous one, which was called The Island of Dr. Marrow. And that one was about the hybrids. Dr. Marrow in an island was doing hybrids between human and non-human. And you see the semi-human creatures running around and crawling around, hybrids, chimeras.
Hybrids of different organs?
Of different mammals. Yeah, he was cloning human embryos into mammals. He was fusing human embryos into mammals. So that's the previous one, older, called The Island of Dr. Marrow.
It's possible.
It's possible. We'll cover that in a few lectures. And the NIH just tried it a few months ago.
It's possible.
Yes, that's why it can be done. Should it be done? It's a bioethical question. Should it be done? Hopefully I've given you enough arguments to say, no, absolutely not. We're killing human embryos. We're manipulating human embryos. We're involving eugenics here. And it should not be done. Okay? So that's when we talk about, going back to this previous slide then, you understand cloning a human is cloning a mammal. Biologically, the technique is the same in the lab. Okay? It involves enucleating an ovum and renucleating that ovum with a somatic nucleus, which makes that ovum revert, in some cases, in the viable cases, revert to a zygote level, and then go forward with development. Whether it's reproductive or therapeutic is simply the end, what is intended, right? In reproductive cloning is to generate a new human being for a couple who can have him, who don't want to have him, who want to experiment with this, etc. But in therapeutic is to obtain embryonic stem cells, ESCs, which means that that embryo has to be killed. So here we have a double offense with therapeutic. in that an embryo is generated in the lab, a human embryo, just to be killed for his or her stem cells. The irony of all this is that the scientific community has spoken very loudly against reproductive cloning and has spoken very favorably for therapeutic cloning. This is the irony. In fact, when you see legislators, let me cap this off with a political thing, because I've been involved with some of this at the state level. A few years, about 12 years ago now, there was an initiative going through the state of Florida for extracting, I think it was $300 million from our state budget for stem cell research and human cloning. But they wanted to do therapeutic cloning, okay? For the therapies, exactly, to obtain the embryonic stem cells. But they, in the bill that was being proposed for Tallahassee, for our state legislature, okay? Because they wanted this from the state budget, $300 million. It said that they were opposed, they were against human cloning. So they say, we are against human cloning. And this bill, the first act of this bill is to ban human cloning officially in the state of Florida. But of course, they were talking about reproductive cloning. But then they said, we want to obtain this money for somatic cell nuclear transfer. Somatic cell nuclear transfer. And that's the word. Let me put it here. I should have. This is the fancy name for cloning. Okay? SCNT. Somatic cell nuclear transfer is a fancy name for cloning. What are you doing? You're taking the nucleus of a somatic cell, you're transferring the nucleus of a somatic cell into, the implication is, into an egg, into an enucleated egg, all right? So somatic cell nuclear transfer, SCNT, is for creating an ovum with a donor nucleus. In other words, for creating a zygote, for generating a zygote. And this is the official, the scientific term for cloning, all right? Somatic cell nuclear transfer. But they use the language, you see, therapeutic cloning. Okay, this is Science Direct. This is a scientific journal. It's a news thing, reports science. And so in that bill that was going, at the state legislation was actually a, what do you call that? They wanted to do an amendment to the Constitution, to the state constitution, to allow for a line, for a budget line, for somatic cell nuclear transfer, for therapeutic somatic cell nuclear transfer. But the first part of the bill was to prohibit human cloning. They're talking about reproductive cloning. See? So it's a manipulation of the words. They're using semantics, they're using the words to confuse the people. They say, no, what we're doing is not human cloning. Human cloning is for the purpose of creating another human being. We don't want to do that. We're against that. What we want to do is we just want to generate stem cells. We want to generate stem cells to cure all your illnesses and all your diseases in the state of Florida, because state of Florida has to go up in the technology and in the clinical trials. And in fact, there is that technology corridor, which for a while was a debate between Orlando and Palm Beach. Eventually, Palm Beach lost out to what they call the technology corridor, which I think is I-4, Orlando to Tampa, Interstate 4. That's a big high-tech corridor, all right? And that's where a lot of money, millions of dollars are being pumped in there to develop that as a high-tech corridor. Mm-hmm.
But I think that is because of their denial that the zygote is human.
Exactly.
It goes right back to that.
It goes,
They don't see,
exactly.
yeah.
They just
they
don't
the
see
correlation
the zygote as fully human.
as fully human.
Okay.
Yeah.
In fact, Bernie Siegel, he's the one who was putting this through the state legislature, or wanted to do this amendment, and got a popular ballot going on throughout the state, and a group of us countered it with another ballot to stop this kind of, to not use our tax money for stem cell research, precisely, for embryonic stem. And well, we tried, and we started developing a campaign, and we started gathering thousands and thousands of petitions, signatures on the petition to stop it. So we started a counter movement, okay, but they were gathering thousands of petitions too. And so we were race to race. And what happened, this was under Bush, on the W, and then the new administration came in, the Obama administration came in, so this must have been in 2008, right? His first time around. And because, oh, okay, so let's get into the politics. Bush must have been about 2001, because I was in Boston preparing to get into graduate school, precisely to study the PhD in genetics. And it was about 2001 that W put a ban on using federal money for creating new embryonic stem cell lines, okay? But he allowed federal money to be used for the existing lines that have been already developed, like University of Wisconsin, Madison, one of the ones who started even with bioethics early on, and they had already developed some embryonic stem cell lines, human embryonic stem cell lines. But to that point, there was very little or no federal money that I know. But W then drew what we call that line in the sand, that he said, okay, from now on, there is a ban to use federal money for generating new human embryonic stem cell lines, or human embryonic stem cell research. Ban on federal money. Doesn't say anything about state money, doesn't say anything about private money, okay? That's still available to the day. So he drew that line in the sand, and he said, well, yes, but federal money, it was like a compromise to get it through Congress. Federal money could be used for the lines that have already been developed. In other words, those embryos already were killed, and now the lines are already developed. We call them offensive lines because they come from killed embryos. But no new human embryos, at least, will be killed with federal money, with our tax dollars, okay? So that was considered a brick for life thing. And that was under his administration, the first term. And I think he did that early on, probably around the March for Life, around January, late January or early February of the inaugural year, as a pro-life statement. Then his administration went through, and so there was that ban. But at the state level, and that's what they were doing, these efforts on the state level, and California had that famous Proposition 72 or something like that, 71, where they extracted, again, about $300 million from the state of California to do Proposition 8, to do stem cell research, human embryonic stem cell research, okay? But all these legislations were going through at the state level, talking about first, they would all start, there was like a pattern to them, they would all start condemning human cloning, which they were talking about reproductive cloning. But then they would go on to solicit the funds for somatic cell nuclear transfer, which was just generating stem cells. Because they don't say that somatic cell nuclear transfer actually is therapeutic human cloning, and those stem cells are embryonic stem cells, okay? And that's the legislation that was going through, and that's what we try to stop here. And so we were there neck to neck, and it was going to come up in the ballot precisely for, so what was President Obama's first term? Was 2008?
Yeah.
Okay.
Right, okay. So this was going on all throughout 2007, I think we started, throughout 2008, because we were building up to, I think we had to have about 20,000 signatures or 10,000, I don't know how many thousands of signatures to get it into the ballot for the November ballot, the presidential ballot, and to be one of those amendments or one of those ballot items, okay, on that ballot, which was a big one because of the presidential race. Well, what happened was that we did not get it into the ballot, but the opposition did not get it into the ballot either. But then with the Obama administration, precisely, I think around January of 2009, right after the inaugural, around the March for Life, President Obama took off the ban, W's ban on federal money with an executive order. President Obama was known for his executive orders, right? Because the Congress was locked in. And so by executive order, he reverted the ban that W had established. Therefore, the flood monies, the floodgates opened from NIH and NSF to do human embryonic stem cell research, in other words, therapeutic cloning, at the federal level. So at that point, the state, it didn't matter because the few millions that they were trying to get from the state, they got many more millions from the feds, okay? And so that's the current state of affairs. Still, as I know, to this day, there's still that, that go ahead. So I think we're kind of waiting for the current administration to secure that ban again.
Well, one of the first acts that President Obama did, and we were there in Washington because that was a week he was in
At the March for Life, yes.
Was he rescinded what Bush had put in on the Mexican policy.
Oh yes, the Mexico City policy.
Yes, yes, the money that we give to like the United Nations.
Yes.
None of that money could be used for abortions in any country that the United Nations will allocate this money to.
Correct.
That was the first thing that President Obama did as the President of the United States.
Yes. Yeah.
So we know how much he supports abortion.
Right. Exactly. So that was, it's known as the Mexico City policy,
yes.
And that was a UN meeting in Mexico City. I believe it was in the 80s. I can't remember exactly.
Bush was the one who put it in and said no more. We're not, no. And
yeah.
Bush signed that thing.
Yes, and it was
the first act that Barack Obama did.
Right. So that ban was at the international level. In other words, that's outside of the national boundaries. So it's beyond NSF and it's beyond NIH. But this is the millions of dollars that the United States gives in international aid, especially poor countries of the world, right, developing countries, and millions of dollars of aid that is given.
But Father, that including China.
Yeah, exactly.
That included China as well.
Yeah, yeah, exactly. Yeah, because if they qualify under UN standards of aid, right, because of poverty level and this and that, then they would qualify for that aid. So you're right. Bush had put in place what is known as the Mexico City policy, which was to not use federal money for international aid that would go toward abortion, okay? And so, like you say, one of the first acts that President Obama did was by executive order to rescind that ban. So, yeah, and I think that now that's what President Trump just did now around the March for Life also. Yeah. He again reinstated the Mexico City policy. That's international policy for the United States. You know that in the United States, it's a little particular. All of the international policy, all of the diplomacy of the U.S. really falls on the executive branch, on the White House, right? Unlike other countries, in many other countries, to be a diplomat is actually a career. You can study political science and you can study for diplomacy and diplomatic schools and so forth in many other countries. But in the US, for some particular reason, diplomatic appointments or foreign appointments are political appointments. In other words, they go by party. So every time that the party changes in the administration, in the White House, all those diplomats, they have to present their resignation, and then it's up to the new administration to either rehire them or appoint new diplomats. That's their political appointments as opposed to career appointments.
In fact, Father, I think we marched that Thursday, and Obama signed it the Friday.
Right. Yes, it was something like that. It was really, it was timed. There was, I mean, these are historical facts, you know, and Obama has served his 2 terms and everything else, but these are historical facts that it seems like the intention was precisely to make a pro-choice statement right around within the 24 hours of the March for Life, when you had hundreds of thousands of people marching in the cruel weather of January that have been there in Washington, D.C., you know, and him from the White House with a brush of the pen release those funds for international aid to abortions.
I will never forget that.
Yeah, it's there. It's there for history. And but this man just did it. Just did the reinstated the policy. Okay, President Trump, right around those days also, because it coincides with the inaugural. The inaugural is always in January.
January 28th.
And well, yeah, I think it's around the mid-January, the inaugural, but it's within 1 or 2 weeks of the March for Life. Okay. Okay. What else? So we've seen stem cell research. We've seen cloning. What's the first thing to do? Distinguish, right? Human, non-human, embryonic, adult, cord blood, cloning, therapeutic, reproductive, but at the end of the day, human cloning, who has a right to generate another human being in the lab, right? To literally manufacture a human being in the lab. There's nothing natural about this process. It is forced in nature to revert a nucleus, a differentiated nucleus to pluripotency. And so it's truly a big offense to nature. We don't have a right to do it, but it can be done. Okay, friends, this is all I have. So I'm ending here. Thanks again. And I wish you a great week. God willing, we'll continue with this next week, okay?
Thank you, Carlos.
So remember your summaries and everything else by midnight of each Wednesday, especially those of you who hold more than one summary. Catch up, please. Okay? Thanks a lot. Many blessings. Okay.